Welcome to the Modern Biopharma Podcast. The world of today's medicines is big, complex, and constantly changing. Our goal is to give you insight into the people that are manufacturing the medicines people need every day, whether it is the people taking care of these facilities, or those building them, or those maintaining the quality and integrity of every dose that is delivered to the patient. We interview the people that are making it happen and give you a chance to get to know them while we're at it. Thanks for saying hi. Hey everyone, welcome to another episode of the Modern Biopharma Podcast. I'm Jesse and I'm here today again with Brian Hagopian. He's a chemist at Clearwater Consulting, and Brian's an industry expert with 30 years high purity water system design, construction, commissioning, and validation experience. Delivered a wide range of solutions to premier pharmaceutical, biotech, semiconductor, research, and other high technology clients. He's a recognized industry leader and active volunteer with a number of professional societies. And I specifically know him from his activity within the International Society for Pharmaceutical Engineering, ISPE, where he is a past president and done a whole bunch of stuff for them over the years. So that's where I know Brian from. And he's lectured on a bunch of topics associated with USP purified water and water for injection and semiconductor grade water. So hi, Brian. How are you doing today?
Hey, great. Happy to be here.
So we're going to be talking about sampling and monitoring in a pharma system today. And I just wanted to sort of ask you, you've been doing this for many years, and we talked about a number of different industries, but I got over 30 years high purity water system design. How has sampling and monitoring changed over 30 years of water system design? You've done a few of these. So what kind of things have changed in 30 years in water systems?
So the first thing that changed in the pharma industry is back, and I believe it was in the late 80s. Don't hold me to that. But up until that point, and dating back all the way to like 1890, there were a number of different attributes. And I want to say there were like a dozen or 15 different attributes that pharmaceutical companies were required to test their water for. And I'm gonna look off to the side to make sure I list everything. Chlorides, sulfates, nitrates, pH, calcium, carbon dioxide, heavy metals, ammonia, bacteria, oxidizable substances. Okay. Now, each of these attributes were things that a quality control laboratory at a pharmaceutical company had the capabilities to test for. And all of this testing involved going to a spot, collecting a sample, bringing it to a laboratory, and then somebody from the quality control department would follow the established procedure, add 3 drops of this, add 2 drops for this, shake it around, wait 5 minutes, hold it up to the light, and look for cloudiness. And those were some of the acceptance criteria. If it wasn't cloudy, it was a pass. Okay, so we had all these different attributes, and all of the, and the chemistries involved were things, with things that every quality control lab could set up and do the testing for. But, in the late 80s, people recognized that, hey, listen, some of these things are antiquated. They're not quantitative, they're qualitative. Is that really cloudy? Or maybe it doesn't look so cloudy. Okay. So they convened an expert committee to try to update the standards. And what they did is they reduced the requirement for all those things down to conductivity, TOC, bacteria, and for injectable grade water, endotoxin. Okay.
Well, that makes it a lot more simple.
It does make it a lot more simple. And in fact, a lot of the other elemental impurities that we were testing for fall now under conductivity. That's why there's a conductivity requirement. Okay, so so we went from having 12 or 13 different attributes to having 3 or 4. And in fact, most of the pharmacopoeias now don't really require bacteria testing, which I find kind of interesting, because that's probably the single most important contaminant that pharmaceutical companies have to make sure when they're making injectable grade water. It's the thing they've got to make sure that they maintain, right? Water is supposed to be sterile. So that's, that was a huge change in sampling. But one of the things that I've noticed is that a lot of the quality control laboratories, even though this change got made, a lot of the quality control laboratories had been, established, and for the last 75 years, samples were brought to them and they did all the testing. So there's been a reluctance to accept online instrumentation for some of these parameters, and we can talk about that a little bit more, when we get into things. But those are the things really that have changed the most over the last number of years.
Interesting. Yeah. So it's kind of funny because in some respects you feel like things have gotten more complicated because we're able to detect at such a higher level than we used to in the past. But in this respect, it actually got simplified because we were able to realize, like, this pretty much is telling you what you need to know. You don't need to know all those other things, because if this is good, then all of those other things, by their very nature, kind of can't exist in the water, right?
Right.
Yeah.
Right.
So that's like, so why do we still test those things? So I still test those things from time to time. You go into facilities and you're going to test for a lot of those constituents sometimes, even in pure, really pure water. So what are you testing those things for?
Well, a number of these things are tested for because of the pharmacopoeia requirement, right? Ultimately, there are a couple of reasons to test within a water system and within a distribution system. The most important part, the part that quality control and quality assurance want to know is and in fact, when the FDA comes in to audit, what they want to know is what's the quality of the water going into the product, right? If the water is used to make a batch of product, they don't care about anything in the water purification system. They don't care about anything in the distribution system. They care about what's coming out the end that's going into the tank that's being used to make product, right? That water. So in as much as you can draw a parallel to what an instrument might measure in a loop and what's coming out the end of the pipe, there's consideration that can be done for accepting an instrument rather than doing sampling. One of the debates I always have with people in quality control is conductivity monitoring. One of the challenges with conductivity monitoring is it changes as soon as you take a sample. Carbon dioxide from the air dissolves right into the water, and it happens pretty quickly. And if you look at the requirements for testing conductivity in accordance with ISPE requirements, there are 3 stages of conductivity, right? If it fails the first stage, then you do something else. And if it fails the second stage, then you do something else. And all of those things are associated with the fact that the water absorbs carbon dioxide from the air and it changes the conductivity. Okay. Now, the conductivity of the water, I'm kind of straying into another area here, but the conductivity of the water is something that doesn't really change throughout a distribution system. So you can measure the conductivity at the supply, you can measure it at the return, and assuming that you've got, you don't have giant dead legs leading from your distribution system to wherever you're filling. If you measure conductivity at point of use, all those readings are going to be the same, because conductivity is an attribute that's uniformly distributed. It's a chemical constituent. Okay.
Right.
And there's justification for being able to use an instrument reading rather than having to sample. You're going to get higher readings when you sample. You're going to get more variation because of the sampling technique. And in fact, back in 2016 and in the 3 or 4 years leading up to 2016, I was fortunate in that I was able to lead a team of 20 or 25 industry experts to produce a sampling guide. One of the major reasons as a consultant that I'd been called in to work with pharmaceutical clients, turns out in 90% of the cases, it's not a water quality issue. It's not a equipment issue, but it's more of a sampling issue. And sampling is an extremely complicated process. And we actually put together a 120-page good practice guide to talk about all the aspects of sampling that you have to consider and that can impact, the quality of the water that you end up getting through the sampling process.
So at a pharma plant, I've watched this. I don't do it much, as more of on the vendor side. I can assist and be there and be part of it, but ultimately, usually the customer's doing it. How many of these guys are getting full training, or I don't know what the level of training is out there in regards to sampling. I know you have that guide. I've read a bunch of it. And when you talk about pure water, it takes almost nothing to mess up. It takes almost nothing to mess up those samples, right? I mean,
well, a part per billion of TOC is an incredibly small, it's 1 grain of sand in a 2 foot by 2 foot by 2 foot container. I mean, that's, it's tiny. It's tiny, tiny. Interestingly, when we first started out to produce this sampling guide, I almost kind of found myself looking in the mirror saying, why would I produce a guide like this? I get called in so many cases to consult with people that have what turn out to be sampling issues. If I help to produce a definitive document, I'm going to be talking myself
out of a job.
Right. I'm going to be talking myself out of a job. And ultimately I said, listen, it's for the good of the industry. That didn't stop me. But, sampling is something that's near and dear to me. And it's a major cause for concern for a lot of pharmaceutical companies, and I see all sorts of things, in different places. Some of it relate to where the sample point physically is located. I mean, you can have all the procedures and documents that you want, but I had a situation at a local client where they were finding bacteria, and we sat down in the conference room and, we looked at the procedures, and everybody swore up and down that, this is exactly the way we do everything. And I said, well, let's go look at that point. And it turned out that in order to get at the sample point, you had to reach around something literally the size of a dishwasher. And if you flushed the sample valve according to the procedure, the only way you could get at it was to literally reach it. And, if you turned it full blast, it's going to shoot you right in the belly button.
Yeah.
Okay. So nobody's going to do that. So then you kind of have the aha moment where everybody says, well, yeah, we really don't use that procedure at this point.
Yeah.
Okay. And now all of a sudden people start to realize that, hey, we got a problem here, and it's the location of the sample point that makes it more challenging. So there are so many things that, that go into this, and people don't usually think about sampling and sample valve location during the design phase. They just, hey, we got to put a sample valve in. It's on the schematic. We let the installer put it in, and nobody thinks about it until late.
Yeah. So it's funny. You mentioned that, like reaching around to sample. So I know you've done more than the pharma industry, and I have as well. And I've seen it in like the labs where they have ASTM type water, and in certain versions of those waters, they have very strict bacteria specs, right? Similar to WIFI. And the thing that I've seen in so many of these places is they have these DI faucets that, they got the little crook neck, and the good ones have flow that goes all the way to the tip and then back, so that these things, so that they don't grow bacteria, right? Cheap ones don't, but most of the good ones do. But then what do all these places do?
Yeah.
They put a tube on the end of it that's like 3 feet long. And it sits there like in its own like water in the bottom of the sink.
Yep.
And I've been called in. I tell you, I've been called into many customers like ripping about, the water's got all this bacteria in it. We sampled it every day for the last week. And you go in and you look at it, it's like a green hose, and it's not a green hose. You're like, well, I think I found your problem. And it's, I get why they use these things for sure. But at the same time, I don't know, maybe it's just because I'm a water guy. It's like, did you look at this? Like, does that happen maybe on the higher ends? I haven't seen it in pharma as much as I've seen it, say, in the healthcare hospital research kind of space.
Yeah, I mean, there can literally be, dozens of different reasons that you can have issues with sampling. Dozens. So, and yeah, that's one of them. That's absolutely one of them. So all the care that you take, if somebody puts that little hose on the end.
Yeah. Or somebody, a lot of these systems say they get a new little area added onto the loop, and they end up with little dead legs. I think that happens a lot of times, especially in older systems where they're retrofit. It's a 30-year-old loop, and you end up with hideout spots that cause tons of problems for them. So, all right, cool. So we've talked about like sort of the things that we're looking for when we sample. How do you prioritize some of these, ways of sampling? How are you prioritizing different constituents? How are you prioritizing where you're sampling? Like some things I know. Here's an example. You might want to, in a pre-treatment system, you might want to be sampling for chlorine right after a step, right after a carbon, because you want to make sure that that's doing its job. So why do we do that if, ultimately down the line, we're going to have a conductivity measurement anyway, right? Why would we measure things along the process if we know that the final step is going to be catching those things anyway?
Well, okay, so you touched on a really important point, and one where we need to make a distinction. We need to kind of draw a line and talk about when we make measurements for what we would describe as process control versus what we would describe as quality control. Quality control being sampling the quality of the water that goes into the product. Okay. Mm-hmm. Whereas process control is, to use your example, if we have a carbon filter, we would sample for free chlorine after that to make sure that the carbon is doing the job that it's doing. Now, free chlorine is not a parameter that's required by USP and the quality control function would never be testing for that. Okay.
Right.
But the engineering and the facilities people want to know, is that carbon filter, if it's there, doing its job? Okay. The same thing with a water softener, right? We would look for calcium and magnesium coming through a water softener, whereas, and again, that's a process control. Is the piece of equipment working? So it's really important when you decide on what you're sampling, it's really important to understand the intention of each component in your system, what it's supposed to be doing, and sampling appropriately for each of those points. People get really nervous about bacteria levels downstream of a carbon filter. And if there's a reverse osmosis process followed by a distillation unit or 2 reverse osmosis processes, I'm going to tell you that you don't need to really worry about the bacteria coming out of the carbon filter so much.
Yeah.
RO is going to take care of it. Whereas, I've seen people, anytime the bacteria count goes up over the level that is accepted by, for drinking water standards, we've got to hit it with hot water, or we've got to steam it, or, we got to do something. And so it's caused a lot of people to turn away from carbon filtration that maybe that decision shouldn't have been made. Mm-hmm. So, to kind of get back to your question, the most important thing is to distinguish between, are we looking for an attribute for process control purposes, or are we doing it for quality control purposes? Okay. Mm-hmm. And the quality control, the quality control for product quality purposes, that falls under the QA and QC folks. Whereas for process control, that falls under the engineering, facilities, and maintenance functions. And interestingly, we could have different limits for process control and quality control, alert and action limits, because frankly, A good process control system will identify a problem and rectify it way before it affects product water quality.
So how
Okay.
does someone differentiate between the two internally and not, like, so say you set up a process control. So, you want your quality control to be less than 1 microsiemen water, say, okay. And your process control is, hey, if it gets above, half a microsiemen, we need to be paying attention, right? That would be an example, I think, of what you're talking about, right? Because if we know it's going over that half, something isn't right, let's make sure we check on it. So how do we sort of keep those divided so that we don't end up causing quality control problems when in reality it hasn't caused one yet?
Well, so it's two different functions, and the process control falls under like a different department.
Mm-hmm.
So, I think, I don't want to say never shall the two meet, but process control should set up their own parameters, their own testing parameters, their own alert and action levels. And, if the requirement is 1.3 microsiemens or less from a quality control standpoint, but a process is capable of making water that's 18 megohm resistivity, well, then that's where process control should start looking at stuff. And if a process is supposed to be making 18 megohm water, and all of a sudden the quality starts to drop to 15, that should be a flag, and they should have an alert to go figure out what's wrong before it impacts product water quality.
Right. So with the new, with a lot of these places going to membrane-based systems, especially a lot of these smaller systems, you're going to now have, a lot higher quality water probably than they've been used to in the past for a water for injection, right? Because I think most
Potentially.
well, on its face they could anyway, because before they were making half a microsiemen, 0.4, something like that out of a still, and now they're, it's coming out at 16 mega ohm right away. So it should be better, and you're way above. And I think in the past, say, still, you're watching some of these numbers and going, maybe there's an issue, and it's a lot closer to the actual quality issue than now with some of these systems. Your TOC should be lower. Everything should be lower in a lot of respects. Do you change your process control parameters that you're watching as a result, or do you just sort of stick with some of the old ones? How do you do that?
No, I think you look at the technologies involved and what they're supposed to do and determine what it is you need to monitor. You only need to monitor, at for example, reverse osmosis unit. You identify the critical process parameters that you've got to measure that will tell you that the reverse osmosis unit's working properly. And those could be the operating pressures, the differential pressure across membranes, from stage to stage. And it's the percent rejection, which is a conductivity measurement.
Mm-hmm.
Okay. And, those are the things that tell you, hey, are my membranes doing what they're supposed to be doing? Or are my membranes getting dirty? Do I need to service them? Et cetera, et cetera. Okay. So it depends on the device and what you establish as the key, critical process parameters that you've got to monitor to tell you that instrument, that device, that treatment process is working the way it's supposed to work.
Mm-hmm. All right. So it's very obvious to me that there's so many different questions and ways we could go with this discussion for sure. And I had all these questions written down. I'm like, oh, I didn't ask like 4 of these still. But one of the things I did, we started to touch on it, and I think we wanted to originally, and I just haven't gotten there. So I want to ask this now, but you talked about how with conductivity is this easy one. Everybody knows it. It's very familiar to everybody. And it's something within water, how it's going to be the same at the beginning of a loop and the end of the loop for the most part. It's consistent throughout. And so we don't have to have, a conductivity probe every 10 feet in a water system. But in many water systems, you have, say 20 or 40 use points on a distribution loop. And some companies are actually taking samples out of each one sometimes every day or every week. So what's the driver of why you would do something like that? And maybe, what kind of things could companies look at and go, you know what, you don't need to do that. And here's sort of a way of approaching why you can pull back, and, you don't have to do every single one. Maybe you do some of them this week and some of them the next week. Like, could you talk to that a little bit?
Yeah, so let me give you my perspective, and this is something that we spend a lot of time talking about in the sampling guide. Okay, so of the four contaminants, and I'm going to talk about WFI grade water contaminants, right? Conductivity, endotoxin, bacteria, and TOC. The contaminants that we would classify as chemical contaminants, and these would be conductivity and TOC. These are things that are uniformly distributed throughout a water system. So you can measure TOC at the beginning of a loop, TOC at the end of the loop, and if the loop's integral, you're gonna kind of get the same numbers. Same thing with conductivity. If it's a biological contaminant, on the other hand, that's a whole different creature.
No pun
Okay,
intended.
it's a whole different matter. It behaves differently, and if you've had any experience with pharmaceutical water systems, you may have no bacteria at 15 use points, and you may have 2 problem use points where bacteria always show up. Okay, that's the issue. So of the 4 constituents that the quality assurance function wants to monitor, right, or needs to monitor to be able to release product, 2 of them are chemical constituents, TOC and conductivity. And in my opinion, through the quality control department can go through a process of establishing the consistency throughout a system so that they can rely on an instrument reading and not have to collect samples. There's definitely resistance because, as I had mentioned earlier, quality control departments have in the past tested. They've collected samples and they've tested for 12 different attributes at each use point, and that's the way they're used to doing business. And kind of breaking that mold, there's going to be some resistance. Okay, even though you can logically talk about how conductivity changes as soon as you take a sample.
Yeah.
People still test for conductivity. People still take samples and test them for conductivity. And that's so I think that TOC and conductivity are the two constituents that are amenable for and suitable for and candidates for instrument monitoring, right? And you don't have to collect all these samples as frequently as people are. Now, for bacteria and endotoxin, those are not uniformly distributed in the system, and consequently, you do have to sample for those everywhere.
Yeah. So what I think we're going to do another episode to talk about that for sure. But that's I've seen that in a lot of places. Again, I think in the pharma systems, they're very well designed, and especially the new ones, they do such a good job that it's not quite as frequent as you might see it in some of the other industries where they're making a PVC system for high spec ASTM water sometimes. And again, it's the same kind of constituent requirements, but they got again PVC joints and elbows to nowhere all over a facility, and you're like, oh well, I think I know where all of our problems are coming from.
Well, and alternatively, if you have a stainless steel system and you feed a subloop that's got a storage tank and a heat exchanger and some other stuff in it, I wouldn't necessarily rely on the conductivity reading from the main loop.
Yeah.
You'd certainly want to see if what that subloop is doing, and have independent monitoring there, and it may be worthwhile to take a sample. So this definitely isn't a one size fits all. It's a more, hey, if it makes sense, you should consider it, because it will reduce workloads, and you'll get better data, and it'll be more consistent, and you can have a higher level of confidence in it.
Yeah. Yeah, and I think I don't know what your experience has been with this, but I've talked to vendors that work with a lot of the instrumentation, and I think there's something to trust your instrumentation. I came from the Air Force, so I have sort of that background in the Air Force, where if you're a pilot, you got to trust your instruments, right? Sometimes you're flying blind, and you got these beautiful instruments that do a lot of the work for you, and you got to trust them. And I don't know. I feel like in sometimes with these systems, people don't trust them, and so they're always second guessing and looking for other ways to do the same thing. I don't know. What do you think about that? Do you see that yourself, or
Now, I
think I'm
think a lot
off
of it,
base there?
no, not at all. I mean, I think a lot of it is just based on the historical thing that, hey, quality control has always done the product release testing. And we've never just relied on a reading from an instrument. What if it's calibration drifts? What if, and they can list 100 different what ifs to justify continuing to test. So, companies have to look at, they've got to go through a risk assessment. And it's a process that, that every pharma company knows how to do, and they've got protocols for doing it. But I do think that's, TOC monitoring and conductivity monitoring are the most likely candidates for instrumentation to play a significant role.
All right. So we're gonna talk about the next one that's coming, right? So TOC used to not be that way, right? You had to take samples, and people didn't trust the instrumentation that was online, but things changed over time, and we've moved to a point where it's well established. Everybody trusts it, and it appears that maybe we're moving in that direction for bacteria. So we're going to talk about that on a separate episode, but here we come, RMM, and see what Brian Hagopian has to say about that. But that's all we got for this today on sampling and monitoring. Thanks so much, Brian, for being on today, and look forward to the next one.
Thank you.
Thanks again for listening to the Modern Biopharma Podcast. Please reach out if there is someone you'd like us to interview or a topic you'd like us to discuss. We will make it happen. And don't forget to like us on iTunes or subscribe on YouTube. It helps us get the word out. See you next time.